Estamos reunidos mais uma vez para a nossa sessão científica semanal do IGM, que ocorre todas as sextas, às 9 horas, agora de forma remota e presencial, então híbrida. Gostaria de lembrar a todos que essa sessão será gravada e será disponibilizada no canal do IGM, no YouTube, a partir da próxima semana. Lembro ainda que todas as câmeras e microfones permanecerão desligados durante a sessão. Solicito que os comentários e dúvidas sejam inseridos no chat, para que possam ser discutidas ao final da apresentação. Hoje nós temos a honra de ter o palestrante, o Dr. Raul Castro, fazer uma breve apresentação aqui do seu currículo. Ele é bioinformata, engenheiro bioinformata, formado pela Universidade de Talca, do Chile, com um PhD em genômica integrativa pela Universidade de Major, do Chile. Sua tese de doutorado foi versada sobre o estudo de RNAs não codificantes em espécies de arqueabactérias, eu acredito, em diferentes temperaturas. Atualmente ele é pesquisador do Programa Institucional de Fomento à Pesquisa, Desenvolvimento e Inovação, PID, da Universidade Tecnológica Metropolitana do Chile, trabalhando como investigador associado no Centro CITEMIX, Centro de Biologia de Sistemas para Estudos de Comunidades Extremófilas de Rejeitos de Mineração. Além disso, a linha de pesquisa está centrada no uso e desenvolvimento de ferramentas computacionais para o estudo de funções de RNA não codificadores, tanto em procariontes quanto em doenças humanas. Ele vai contar um pouco hoje sobre isso para a gente. Você tem, Raul, primeiramente agradecer muito a sua presença e dizer também que o Raul ele está integrante, ele é um dos professores que está envolvidos na ação do curso de sequenciamento de células únicas que está sendo ministrada, que termina hoje ou amanhã, eu acredito, hoje, né? Hoje, ontem, terminou ontem, na realidade, aqui no IGM, que ocorreu nas 2 últimas semanas. Então, Raul, muito obrigado, você tem 40, 45 minutos, e depois a gente inicia a parte das perguntas. A palavra está com você.
Bom dia a todos. Vou tirar a apresentação. Vou compartilhar primeiro para aqui. Eu faço isso. Aí. Já estou agora ver? Sim. Já. Bom dia para todos. Eu posso falar só um pouco de português, mas vou fazer minha palestra em inglês, because I don't have many practice in Portuguese. I don't have good pronunciation in English, but if you can understand something, you can ask me at the end of the presentation in English, in Portuguese, Español, if you want. As Leo said, I am a bioinformatics engineer. In Chile, we have this amazing undergraduate career when you study from bioinformatics from the undergraduate studies, and then I perform my PhD in integrative genomics. I have the opportunity, when I start my postdoc in Universidad de Chile, I take a position in the Universidad Tecnológica Metropolitana in Chile also. And now I am in my PhD, and I am an academic researcher in UTEM, Universidad Tecnológica Metropolitana. So today I will talk for you these amazing molecules that are non-coding RNAs. They are just a messenger RNA, and now we know that these molecules have a lot of biological function, not only a messenger RNA, and we know that the 70 to 80% of the human genome is transcribed to a RNA, and only the 3% of the genome is protein coding genes. It's important to take into account that if you are studying poly A plus transit, only the 40% of the genome is transcribed. There are a lot of transcript that doesn't have a poly A at the 3' end. It's important to take into account when you are working with transcript. Also, these molecules are associated with cancer, immune disorders, neurodegenerative diseases, and a lot of disease biological process in human, in eukaryotic, in bacteria, and also in archaea. They are very interesting. Here I show you a review when the authors show they are Brazilian researchers. They show a lot of long-non-coding RNA involved in the pathogenic infection and also in the vaccine immunity when we are infected with pathogen. It's a very interesting review. I recommend to read that if you are interested in non-coding RNAs. And also, they are involved in the pathogenic infection. For example, the helminth can export the microRNAs because it is a eukaryotic pathogen. And these microRNAs regulate the transcription of our cells, and they control us by exporting this type of non-coding RNAs. This is the last biological insight that I want to show you, but it's very controversial because it's, they hypothesize that the RNA exported by bacteria can regulate the host messenger RNA, can regulate the transcriptional factors of transcriptional genes in the eukaryotic cells. And we have a almost accepted publication in this type of regulation, and I will show you later. Where we can find or where we can find non-coding RNA, there are a lot of databases as protein coding genes, but the biggest one is the RNA Center. This is a consortium maintained by NCBI and that kind of consortium. But we have a Latin American database called NRDR, that is a Brazilian guy that published this database. And now we are working to reduce the redundancy in this database, collapsing all the sequences that are 100% identical, in not only in the same organism. Between organisms, there are a lot of sequences that are equal. So we are working on that. We don't have the publication yet, but we create a front-end page when you can access to the non-coding RNA in searching via organisms, searching via a particular RNA class if you want. And also you can explore the data in a taxonomical level if you want to retrieve all the non-coding RNA annotated in eukaryota on a particular gene or a particular species. You can explore in an easy way. But we are working to improve this page. Also, we provide the information for a particular RNA. You can access to the distinct classes that this RNA has, and also where database contain this sequence. And using this sequence, you can align known RNA sequence to a genome of interest. For example, a bacteria that you sequence in your lab. You want to identify non-coding RNA in that genome, you can use the widely used tools to align this sequence, both side high, side blast, for example, and you can annotate the non-coding RNA in your genome. But sometimes the sequence primary conservation are not conserved, and the structures are conserved. The secondary structure of RNA is represented as I show it here in parentheses and dots. And using that information, we create a web server in where you can annotate automatically the transcript that you provide to the server, and also if you upload a genome, annotate the non-coding RNA in all the genome. It's a very nice tool. We are working on a second version of this tool to increase the limitations, decrease the limitations. The RNA families are a sequence that have a lot of conservation in secondary structure and are maintained by RFAM. It's the same like PFAM. It's a database that maintains RNA families. We use that database and make the annotation. This is the job launcher. When you upload the sequence, select the method that you want to use to annotate the non-coding RNA, the filters, and we have these limitations because if you upload a sequence more than 10 mega bases, this take all of our server and crash it. We are working to improve that. And if you want to do with a eukaryotic genome, for example, you can download the standalone version, but it requires a lot of bioinformatics skills to do that. And the results are like this. When you upload a genome, I think this is the sample for the I think. Here they are the RNAs annotated, the RNA family, the code for RFAM, the name of the RNA, and also the location into the genome. And we provide a secondary structure to be compared to the structure from the family, if you want. Also, we are developing this tool that we are not published yet, but it's an interesting toolkit, a Fiocruz script that you can call in command line to identify and annotate and perform the differential expression analysis of the smaller RNAs using any RNA-seq data. We tested mainly in bacteria because the genome is smaller, and the tool aligns the grid to the reference genome, perform the collapse in this grid, and annotate all the regions in a genome with that have transcriptional evidence. You can compare this region to the coding region and also identify the new non-coding RNA or coding RNAs. There are a lot of tools in the literature to perform this task, but this is an amazing fact that I see the last year. It published by a collaborator in France, but she is a Chilean research, and it performs annotation of the smaller RNA without a reference. Perform alignment between the reads and try to find a smaller RNA in RNA-seq data. I use this tool to the bacterial data that I have, and it performs very well. We identify a lot of small RNA from bacteria and human in the same sample. There are other tools that are improved to long non-coding RNA. The methodology is almost the same to this tool, but it is improved to the long non-coding RNA, not for smaller RNA. We now have a very diversity of tools to implement or to annotate non-coding RNAs. And when you have a transcript or a transcript of analysis and you don't know if your sequence is a coding or not coding RNA, you can predict the coding potential. There are a lot of tools to do that, but we implement a tool based on machine learning approaches to identify if a sequence is a coding or non-coding sequence. We publish it in 2021, but here to do this research, we need to implement a lot of reset a lot of the organisms because we have to do one model for each organism. it takes a lot of time to do that, but we only have models to eukaryotic organisms. And we are working to implement or to perform and create the models to bacteria. You only need to have the sequence in FASTA format to provide to the web server and select which model you want to use to annotate non-coding RNA. And the tool perform the annotation. We test for the prediction models. We test all the available machine learning approaches, and the random forest was the best. And then compared to the most widely used, sorry, I don't have control for that. The most widely used tools, and our result was much better than the previous published tool. Only in one of them we have the same accuracy. And this is the tool. You only need to upload your FASTA file with your transcript and select the organism here. And then you obtain a table with all the sequence and a column that says is coding, non-coding, and the score. You can also download the FASTA file for coding genes, the FASTA file for non-coding genes, and perform downstream analysis. It's a very nice tool and easy to use to predict coding potential. So we have database to analyze sequence primarity searches. We have a tool for secondary structure. We have a tool to annotate non-coding based on machine learning approaches. We are working to have a tool to annotate a small RNA based on grids. And what happens if you don't have primary sequence conservation, secondary structural conservation, but you have a functional conservation? There are a lot of RNA that are conserved between organisms, but don't have any conservation in sequence and structure. You can do a widely used approach that is called co-expression analysis, when you analyze the expression value for non-coding and coding genes together and find models of genes that are highly co-expressed between them. And you can select models that have a lot of, in this case, non-coding RNA, and see the functional enrichment of that model. In this case, it's spermatogenesis, and that can see that non-coding RNA have that function, and you can study more in deep. For that, we implement a semi-tool because the co-expression analysis is very hard to do. It requires a lot of bioinformatics knowledge, and we first developed the package in R, maintained by Bioconductor. You can download and perform your analysis in R. And 1 year later, our collaborators published the web server. And I don't participate in this. I recommend to reference this publication. It's the package when we explain all the math between the analysis. And you can submit your expression data. That is a table with the count for each gene and each sample. You need to have more than 20 samples to do a good co-expression analysis because the statistical power decreased a lot. And you can provide 2 optional files. The first one is the interaction, protein body interaction, for example. You can also provide a gene-set file. A gene-set file is a pathway, for example, gene ontology, and then this file contains all the genes that are involved in one particular pathway and all the pathways for a particular organism. And the tool perform in just one line if you using R, or just click on run in the web server and perform all of this analysis and give you the information co-expression model. If you provide a protein-protein interaction, how the proteins are interacting with the non-annotated gene in this. It can be London coding on new coding genes, and you can explore the data in an easy way. we now are working on how to select differential expressed genes. In most of the case, you put a cutoff of 2-fold change, for example, all the genes that are differential expressed more than 2, you select the best differential expressed gene. But we are working to don't do that because you are human. You select the arbitrary cutoff that you want, but in our case, we provide a rank of genes based on fold change, p-value, and mean expression. That's the three values that you have to take in mind to decide the number of differential expression genes that you have. We are working in this tool. You provide a table with differential expression analysis, and there are other input, the gene set file that I talked before. We provide 2 gene sets, gene ontology and reactology. They are widely used, and also you can provide a custom file. This tool is more useful for coding genes, but because the pathway on gene ontology are well represented for coding genes. Here you can obtain the number of pathways that are consistently enriched wherever you select the number of the top differential expression genes. And when you select a few number of differential expression genes, there are less pathways that you obtain as differential expressed. You obtain pathway enriched by upregulated genes and for downregulated genes. And I use this tool to non-code RNA. Before I want to explain the gene set file, in the first column you have pathway, a gene ontology, and in the next column you have all the genes that are reported to be involved in that pathway or gene ontology. And I create a custom file, and I put here a microRNA, and in the next column I put all the genes that are validated to be target of that microRNA. So here I create a gene set for microRNA targets, and I use that gene to select in colorectal cancer the top differential expressed genes with the tool, and the tool tell me which microRNAs are enriched to regulate that those RNAs, and then search in heart failure patient, in the blood of heart failure patient, which microRNAs are overexpressed, because we have the hypothesis that microRNAs that are released in heart failure can increase the proliferation in colorectal cancer. There are some publication on that, but based on the protein. We are working now in microRNAs. Sorry. Just sí. My postdoc project is based on microRNAs that regulate that are released in heart failure and regulate the colon, colorectal colon, cancer. Sorry. The last aim will be performed with a collaboration in Universidad de Chile. Some of the last tools that we developed is MetaVolcano. It performs differential expression meta-analysis. What does mean? When you are analyzing a lot of data sets, each of these squares are a differential expression analysis performed by other authors. You download the data set, you perform the differential expression, but you have differential expression here, and here. Usually you manually try to find which genes are consistently differential expressed in each dataset. to find the master regulator genes for that process. That can be cancer, colorectal cancer, for example. Another study involved colorectal cancer, another. And this tool takes these matrices of differential expressor genes and perform the meta-analysis for you. It gives the number of genes that are consistently upregulated, downregulated, and the genes that are unperturbed by the condition. And also, it's interesting to see when you provide a lot of data sets, when you have more than 4 or 5 data sets, the number of differential expressive genes are stable. We are working on publishing this tool, but we have a lot of problems with the front end and back end. But the tool is work maybe the next at the end of this year. Well, we develop a lot of tools. We use that in many organisms. We have publications in human, mice, rat, plants, and also in Archaea. That was my thesis, my PhD project. And we publish a lot in many organisms. I want to present you this because it is a Chilean native maize that called is UTEM for the Valle, the Utah Valley. And it's very interesting because we detect non-coded RNAs that are involved in the salt stress in this amazing maize because it's very tall, it's a very good maize. And also in human diseases, I work on cancer, work in neurological diseases like ALS. It's an amazing collaboration that takes us 4 years to perform all the analysis and wet lab experiments. And also in Leishmania brasiliensis, for example, we create a database that is down at the moment because we are working to improve this to all the Leishmania species, to annotate the non-coding RNA and coding genes in Leishmania species. Also in pathogens for fish, because in Chile, the fish farms are very important, and we annotate non-coding RNA in this pathogen. And as I mentioned, we have a almost accepted paper research involving the regulation of bacterial small RNA in a small sample of patients with high and low cholesterol. Our finding is amazing because we found a small RNA that regulates all the genes that are involved in the synthesis or degradation of cholesterol. And this small RNA is from this species, and this species are reported probiotic bacteria that is used to decrease the fatty acid in the patients. It's an amazing finding, and we hope that it will be accepted. And the last topic in my talk, I want to present the cell-transcriptomic analysis. I am here for that, explaining how to do that in our computers, explain the practical side. I think that Vinicius Maracaya present you this type of analysis, all the web lab and things that you can do. I explain a little bit. In this kind of analysis, you can obtain the expression value for each cell in a tissue, in a complete organism also. This is amazing because in the classical RNA-seq data, you have your tissue in a tube. You can obtain the cellular types that are involved in that tissue. In the case of single cell genomics, you obtain all the cells that are involved in that tissue and compare the communication between cells. Very amazing tool. And also we have the spatial transcriptomics when you have the location of each cell in the tissue. This method is amazing. It's called the method of the year in 2 years. And this tool can be applied to non-coding RNA. This is my research aim 2 years ago. I worked in how to annotate on or work in non-coding RNA in single cell data, because the RNA are forgotten molecules in this kind of analysis. The interesting thing is only when I see a publication in single cell experiment, they perform the typical things. Annotate the cell, annotate the cell types, annotate the gene markers, cell trajectory, and differential expression, but they forgot the interesting things that are here, the gold mine for me, because they don't perform in the publication cell communication, for example, some do. But in this case, I don't see a lot of publication working on long non-coded RNA in single cell analysis. There are some 3 or 4 papers focused in that kind of molecule. To finish my talk, I want to, thanks to the funding, my postdoc. I ended my postdoc. I'm a researcher. When I started postdoc, I become a postdoc researcher and also academic professor. With a lot of tasks to do, but I ended my postdoc and also participate, as Leonardo said, in a project that studied the extramolecular communities in mining tailings. It's amazing because they have a lot of money to study the function of bacteria that perform the degradation of metals. But I am interested in studying non-coding RNA in the genomes that they are sequencing. And now I'm writing a project to obtain funding to do that. I have to say thank you for your attention, and if you have any questions, you can do in English, or Spanish, Portuguese, wherever you want. Thank you very much.
That's okay. Para de compartilhar.
Não foi um dia.
Pergunta para a Vicky, por favor. Raul, não. Muito gosto. Eu professor sorry. My questions are very basic. I'm not reading about the non-coding RNA. I'm not happy with it. What about the size, the range of size? Are they very short? They are long? Do you have information viruses can also transcribe non-coding RNA? And the third question is, are they located somewhere in the genome, like in the motor, or they are randomly?
Yeah, the first question was the size of the non-coding RNA to the people that are in the Zoom. They have a lot of sizes between the smaller ones, I think it's 15. That is small, and they are a lot bigger, long non-coding RNAs. And in humans, for example, we have 100 kilobases long non-coding RNA with exons, which is a crazy thing because they are non-coding. So, why they have exon and intron? The distribution into the genome, they are located in both strands, positive, negative, in intron, in exon, in promoters. They are located in any part of the genome. And if viruses have non-coding RNA, we are studying viruses, but have a few amount of non-coding RNA, but there are megaviruses that have a lot of microRNA, long non-coding RNA, that have anything that you think to regulate the host.
Bom, eu tenho uma pergunta. Obrigado, Raul, pela excelente apresentação, várias ferramentas, né? Assim, talvez eu seja uma pergunta um pouco naive, talvez, mas acho até para a audiência, para o público que está assistindo. Eu não sei se esse número existe, fechado, mas qual a porcentagem de regulação, tudo que é regulado na célula, alguns sistemas biológicos, seria derivado desses RNAs longos não codificantes ou microRNA, só para ter ideia do impacto que isso tem nos processos biológicos e em regulação gênica, assim, então, principalmente para os alunos entenderem a magnitude disso e como a gente talvez esteja ainda iniciando nos primeiros, assim, iniciando ainda o entendimento desse processo.
Muito obrigado pela pergunta. I answer in English because my Portuguese is not very good. I think my English is better. I think non-coding RNA are involved in anything. I think all the coding genes that we have in humans are regulated almost by one microRNA, for example. All the microRNAs have, one microRNA can have more than 500 targets. This is amazing because we have 2,000 microRNAs, annotated microRNAs, and if you multiply by 500, there's a lot of regulation. And in the bacteria, the non-coding RNA, they take part of 5 to 10% of the genome, and it regulates all the coding genes and any process in transcriptional process, in translation, in location. They act as chaperones. In any process that you know, they are involved in non-coding RNA.
Obrigado. Então, acho que fica aí uma mensagem de todo mundo que está estudando regulação gênica, a importância de dar uma olhada nessa classe de moléculas, que nem é tão nova. Mais uma pergunta, ou do chat, ou aqui da audiência? Eu tenho mais uma, assim, se não tiver, na verdade. Então, assim, se eu entendi, vou falar em português devagar também, mas se eu entendi a proposta para você tentar analisar dados de expressão gênica de RNAs, RNAs mensageiros, junta e tentar identificar potenciais microRNAs ou RNAs não codificadores que estão nessa via, seria a análise de módulos de coexpressão, então, que você mostrou com o semi-tool. Teria outra ferramenta, ou essa ainda é a melhor estratégia para você tentar identificar dos seus transcritos mensageiros, quais RNAs potenciais não codificadores ou microRNAs estariam associados com essa análise? E outra pergunta é se existem pathways, vias, já integrando esses dados, porque algumas das ferramentas que a gente usa, esses dados não estão integrados. Eu disse que você mostrou algumas ferramentas que tentam montar novos pathways, porque se esses RNAs longos, não codificadores, estão coexpressos com RNAs mensageiros, ou microRNAs no mesmo módulo, vocês têm uma proposta de montar esses pathways, ou como é que isso está em termos da literatura de anotação, porque a gente sabe que tem reactome, a gente sabe que tem outras, essas análises de enriquecimento de vias, de inset enrichment analysis, por exemplo. Então, minha pergunta é mais nesse sentido. Ficou claro? Sim.
The first question was
semi-tool and how
yeah. In the case of which kind of analysis you can do, semi-tools or co-expression analysis is widely recommended by non-coding RNA. When you have a small RNAs, the expression value are biased because they are too short and the expression value are too high. In that case, when you are working with a small RNA, I recommend to use target prediction with tools to predict target to correlate or associate the small RNA to messenger RNAs. And the second question was the databases that are focused in non-coding RNA. Mainly the RFAM has pathway or gene ontologies for non-coding RNA. There are not many databases to have this information. It's only maintained by RFAM, and I think RNA Central includes the last year the information on non-coding RNA ontologies, but they are based on the coding gene that they regulate. It's a bias in the information, but there are a few databases.
Ok, obrigado. Mais alguma pergunta? Da audiência? Luciano.
Do you know if any non-coding RNA is being targeted by the pharmaceutical industry to solve any problem?
The pharmaceutical industry don't care about non-coding RNA because they are difficult to work in laboratory to validate the expression to perform experimental analysis to do the lab things, but in many cases are involved in, for example, in leishmania resiliency. Not in resiliency, in another leishmania species, I don't remember. There are a pharmaceutical company that start working with non-coding that regulate receptors that allow to the pathogen to block that receptor and don't capture the drug. There are a few cases of companies starting to work on that I know.
Talvez microRNAs esteja pouco mais avançado. Mais alguma pergunta? Eu tenho uma última, na realidade, sobre a ferramenta que você mostrou, acho que foi construída junto com o Arthur, né, o PEBA, né? Ela está disponível, eu vi que ela está totalmente disponível ou não?
No, ainda não. We are working on improve or fix some bugs in the web page, and we have the first round of review, and they asked to us perform validation of different datasets. We are working to use our tool in different datasets and publish, including the publication.
I was wondering if PEBA was developed by someone from Bahia, because this word is from the Bahianese. Do you know the meaning of PEBA?
No, the Brazilian guys think
PEBA for us is something ordinary, not special.
I think the Elder Nakaya put that name. I don't know why.
Ok. So, se nós não tivermos mais perguntas, então... a Patrícia quer dar uma palavrinha. Você quer vir aqui? Mas aqui o áudio capta melhor aqui.
Só gostaria de agradecer, né, a equipe toda, e Raul e todos que estiveram aqui organizando esse evento. Eu estou impressionada com a complexidade dessa nova área e a quantidade de ferramentas, né, de tools que podem ser usados. Esses outros já são disponíveis, né, na verdade, para utilização. Então, é impressionante. Queria parabenizar. E você já tem um cargo definitivo lá, né? Saiu do pós-doc e já está trabalhando definitivamente na área. Então parabéns e obrigada mais uma vez.
Muito bom.
Obrigado, Raul. Então, vou agradecer a presença de todos. Acho que a gente não tem mais nenhum recado, né? E até a semana que vem, então. Espero vocês aqui no auditório, ou quem não puder comparecer remotamente. Ok? Um grande abraço, um bom final de semana e até mais.